So we are supporting to do research to all faculties and all academics, not only academics, so for our students. So then we increase student research fund from 5,000 to 10,000 from 2025. So therefore, our objective is to inculcate research culture within the faculty and within the university, and also do more collaborative work with other relevant industries and institutions. So they are sponsoring for the conference by National Science Foundation, and also. We mainly for encourage our academics and our students to do more collaborative research, interdisciplinary and transdisciplinary research. So that is a must, because so then the individual research has no value right now. So therefore we have to do research with industry and other disciplines. Then only so we can transmit or then transform our research finding to the society. So in our university, we have a research council. So then all the research grant and everything is governed by and monitored and evaluate by the research council. It is a very good example for the country as well. So then I remember the COP also appreciated our research council in 20 the 3 years before. So then through that model, so we encourage all discipline of our research. No, and then so we have a UBL and also Innovation, Invention and Venture Creation Council. So then we take our research find into the UBL for the patenting and other thing, and then the commercialization through the university business linkage and Innovation, Invention and Venture Creation Council. So far, according to the statistics, Jayawardhanapura has the highest patent filing in the country, in the national universities, and also the commercialized product. All these product and patents, the outcome of our research. So then I strongly believe today is the very productive day for all students. So take this as a good opportunity to the to show your research finding, and share your knowledge and your research results with the others and the stakeholders and other. Then I want to say, then you may get the positive as well as the negative feedback. Take all these feedback. So this Thank the faculty, and very especially I want to thank the dean of the faculty and organizing both the conference, and also all members of the faculty and the reviewers and all the supporters to take this conference as a real conference in Kerala. So with this note, uh, so then I wish you a very good day and share your research findings with the stakeholders and other relevant parties, and get their feedback into your research and your personal career. Thank you very much. With that, I want to excuse you because I have another the foreign delegation in the university. So therefore, I have to be. Thank you.
Thank you, sir. Organized for the first time in the history of the Faculty of Urban and Aquatic Bioresources, University of Sri Jayawardenepura, Annual Research Conference on Bioresources 2026 marks the beginning of a beautiful scientific journey where research findings will become policy implementations in Sri Lanka and around the world. To hand over the conference proceedings to Senior Professor Patmalal M. Manage, Vice Chancellor of the University of Sri Jayawardenepura, I cordially invite Senior Professor Pradeep Javera, Dean of the Faculty of Urban and Aquatic Bioresources, and the conference chair. Thank you, sir. We now move on to the most anticipated segment of today's program, the keynote. This session will be chaired by Professor Surani Chaturika, the conference coordinator of the annual research conference on bioresources 2023. We invite all participants to engage actively with the ideas and discussions presented during the session as we explore emerging developments in bioresources research.
Thank you, Ritik. It's a great privilege to welcome Professor James F. White of Rutgers University, USA, as one of the keynote speaker of the annual research conference on bioresources. Professor White is an internationally recognized plant biologist, mycologist, and pioneer in the field of plant-microbe interactions. His groundbreaking research on epiphytic microorganisms, plant microbiomes, and biological nitrogen acquisition has significantly advanced our understanding of sustainable crop production and plant health. His research has contributed to development of innovative approaches for enhancing crop nutrition, stress tolerance, and plant health through beneficial microbial associations. Over his distinguished career, Professor White has published more than 800 scientific papers and book chapters, accumulating over 20,000 citations and achieving an H index exceeding 80. He is widely regarded as one of the world's leading research in plant science and agronomy, and his pioneering contributions continue to inspire scientists worldwide while providing innovative solutions for sustainable agriculture and environmental resilience. I cordially invite Professor James White to start your talk. I cordially invite.
Thank you so much. I'm very happy to be here at your first Bioresources Symposium. And I must tell you that this is my first time in Sri Lanka. And I'm very impressed with what I see. And I'm very impressed with what I see at your university. And have one of my graduate students is actually from this university. So she is my main connection to your university. And so she brings me here. Okay, so do we have, we have, good. So what I'm going to, okay, it's very appropriate that I'm talking about uh, these microbes in plants uh, at, at your Bioresources Symposium, because this is really a hidden source of bioresources for agriculture and for medicine. The world over, people are studying these microbes. And let me see, I'm going to have to go back for a second. What did I say the title was? Okay, the roles of soil microbes in crop production. Okay, that's a very broad title. But actually, what I'm going to talk about are the soil microbes that go. the bacterium that is inside the plant cells, the root cells. Okay, so plants, if you consider a root, for example, a seed germinates, the root comes out. That root then will begin to secrete exudates, chemicals, sugars, organic acids into the soils, and those will attract the microbes to the root. And then at the root tip in the meristems, the plant will take those microbes into the root cells. And then the plant will take them in as endophytes and begin to process them, begin to take nutrients out of them. So uh, it's a principle that plants will cultivate microbes in their meristematic or their growing tissues. Okay, all plants do it. It's not just it's not just a few. It's not an exception, but every single plant will do this. And okay, this actually shows a root. Okay, you see the root here, and you can see the root hairs coming out, and you see the root tip. The meristem is here. This is blue coloration. The blue is where the microbes are going. The microbes are being cultivated there. The plant is putting those sugars, those exudates, to cultivate those microbes there, and is taking them into the cells of the root. So as that plant develops, it takes more microbes in from the soil. Okay, so why do plants need rich, microbial rich soils? It's because they take those microbes in. They need those microbes for their own development, for their own nutrition. Okay, so this actually shows two roots. And you can see a root of a grass here, Poa pratensis, very common grass for turf grasses in the United States. Okay, used in Europe as well, but it's a cool season grass. What we did is we took the seed and we removed the microbes off of the seed, because these microbes are vectored on the seeds. In some plants, they're on the surfaces of the seeds, and in these grasses, they're on the seed surfaces. Okay, so we essentially cleaned the seed using simple Clorox bleach, right, which is very potent a killing agent for microbes, and then we germinated them. And when you germinate them like this, you can see the seedling works. The root comes out. The root looks fine. Okay, you can see the root cap there and the cells coming off. You don't see anything on it though, and you also don't see any root hairs. There's no hairs forming. Okay, whereas here you see we did the same thing. We took the Clorox, we cleaned the seed, but then we added microbes back to it. And when we add the microbes back to it, they're mostly bacteria. Now you can see the microbes, they're stained here, the brown color, those are the bacteria there. And you can see there are hairs and little dots out there. Those are the bacteria out there. The the plant is taking in those microbes, putting them in the cells, and then it's triggering hair development, root development. Roots grow. The plant actually requires those microbes in order to develop properly. It doesn't form, for example, it will not form root hairs. The roots won't branch. Roots will be very tiny. Plant without the microbes, plants cannot be plants. They can't grow properly. They can't grow properly. They can't get nutrients properly. Okay, so it's critical. I mean, these soil microbes are very important. Soil and also seed microbes, also microbes in the tissues of the plants, the endophytes. They're an important bioresource. It happens also in shoots of plants. So here we can see this is actually a moss plant, and you can see the leaves developing here, and you can see the shoot here, and where the meristem is in here, and you can see this little. Those are the bacteria that the plant is cultivating on the shoot. It's growing those microbes there. And it's taking those microbes into the cells. Shows the moss leaves. And you can see the cells here, and you see the little dots in there. Those are the bacteria that have been taken into the cells of the plant. So they're actually becoming a part of the plant. They're being incorporated into the plant. Okay, this is just an image that shows where we've lettuce. One of my graduate students did this image where she took the bacterium, and this happens to be I think it's a pseudomonad bacterium. She tagged it with a fluorescent, in this case, a red fluorescent protein, and then put it into the plant and then used confocal microscopy to visualize it inside the plant. So she tagged it essentially with this red fluorescent protein. And now you can see the red. Those actually are the bacteria that are inside the lettuce root, or rather, I think these are leaves actually, leaf cells, because you can see a, a, a stoma there, guard cells there. All the red are the bacteria that were taken in. So they are taking these bacteria into their tissues, using them for development. This shows again, you can see the bacteria on the outside, stained red, and you can see them inside the cells. And you can actually see, in some cases, you can see pairs where they've recently divided. So they're actually living inside those cells of the plant. Okay, so why are plants doing this? Why are they taking in the microbes? They're taking them in because they need the microbes for development. Those microbes are producing hormones. They're interacting with the plant, and they're causing the plant to grow and develop. They're also turning genes on in the plant. They're making genes more stress hardy. They're making them resistant to reactive oxygen and oxidative stress, to even to herbicides. They're causing herbicide resistance through this oxidative mechanism. And they also are being used by the plant as a source of nutrients. Plants are getting nutrients out of these microbes. And when we put different microbes onto plants, we see that the plants absorb different kinds of nutrients. So different microbes deliver different nutrients to the plant. Okay, so the nutrient extraction, one of the process now that I'm going to talk about is called rhizophagy. Rhizophagy means rhizo for root, phagy for eating, root eating. And this is the process where plants take in the microbes and degrade the microbes as a source of nutrients. Another process that I'll mention that is not as studied is this nitrosomes, the nitrosome. And the nitrosome is simply a vesicle in the plant where nitrogen fixing microbes accumulate, and some nitrogen is fixed in those vesicles. Okay, and this is, all plants can produce this. It's not as efficient as, say, the legume nodules, right? The root nodules, the rhizobia, those are very efficient. This is less efficient and more temporary process, but it delivers some nitrogen to the plant. And so plants in nature will be doing this process. They'll be taking the microbes in to get nutrients. Okay, just to say a very little bit tiny bit about the nitrosomes. We don't know a lot about them. But basically, you have a vesicle. Nitrosomes are these structures where plant develops a vesicle, and nitrogen fixing diazotrophs, bacteria, are accumulated in the vesicle. They don't last a long time. They're taken in at the root tips, or they're taken in at the shoot tips, and they will eventually stop working as the plant grows. So they're only in the growing tissues, in the rapidly growing tissues. Okay, and some literature that refers to these nitrosomes, that you can see here in these papers that I've indicated there, those references. There's not a lot known about them. One of my graduate students a few years ago did some work with cotton, in which she used a bacterium, in this case, a nitrogen-fixing bacterium. She put it onto the plant, into the cotton plant, and then she assessed what genes were turned on in the plant. And the genes that get turned on, you can see this nitrate metabolism, nitrate assimilation, and the cellular nitrogen compound metabolic process, among other genes, get turned on when those microbes go in. And we think that's the indication of that nitrogen that's coming out of these microbes or coming as a result of the microbes being absorbed into the plant. What are these microbes? These microbes are soil microbes. They all come from the soil. Everything we isolate from the plants actually are coming from the soil. Now, they will go onto the seeds. And so when you get a seed, that seed will carry not only the embryo and the food for that embryonic plant, but it'll carry the microbes with it. Okay, so we have to be careful when we sterilize seeds because we get rid of some very beneficial and important microbes when we do that. Uh, and so those microbes are seed vectored, but all initially from the soil. Okay, this shows a germinating seed. And you can see the root coming out. There's a little bit of yellow coloration around here. Those are the bacteria. There are small communities of microbes on seeds. And of course, they're important for the plant. Tomato, a plant like tomato, is you'd think they would be sterile. Okay, tomato, you cut into the fruit, you have the seeds in there. You would think they would be sterile in there, but they're not. In fact, there are bacteria in those seeds. Oops, went back. If you were to cut a seed, we can cut these seeds and look at it with a microscope, and you see biofilms. We see biofilms of bacteria around that embryo. So there are bacteria that are inside the seeds as well. The tissues around the seed bear bacteria as well. So they are carrying bacteria inside and on the surfaces. So we have to be careful with any kind of sterilization of seeds because that removes some microbes that are very important. So seed sterilization is definitely a process that's not good. On the other hand, it's also possible to take some of these microbes and put them on the seeds. Okay, the biostimulants. And so there's a whole industry for patenting these microbes, for getting these microbes out of the soil, for putting them on plants and exploring how they promote the plant growth and protect the plant from disease and so forth. Um, I'm going to talk now a little bit about this rhizophagy process. Again, rhizo for root, phagy for eating. And this process was actually discovered by some Australians. And these are some of the people who did this. These two people, in addition to a whole other lab group that worked on this. But what they found is that plants are taking the microbes into the roots. And what they did is they took yeast and they took also a bacterium, E. coli. They labeled it with green fluorescent protein and they fed it to plants. They fed it to Arabidopsis. They fed it to tomato. And then they used their confocal microscopy, fluorescent microscopy, to see those microbes inside the plant tissues and cells. And they showed here, for example, you can see a root hair. You see the little green dots. Those are the yeast actually inside the root hair. Okay, and they saw here too. You can see all those. Those are bacteria inside root hairs, inside root cells. And what they showed is that in time, those bacteria disappeared. Okay, and so they proposed that this is a nutritional process and that the plant was actually degrading or eating. They named it rhizophagy, so root eating, that they're eating these microbes, that they're taking them into their tissues. It's not as simple as that, but that's essentially what is happening, the basic process. This shows some bacteria, and you can see them inside the root hair there. You can visualize them now. If you don't use a microscope, you certainly can't see them. But even with the microscope, they're difficult to see. The way that we see them is we use reactive oxygen stains because between the plant and the microbe, the plant is producing reactive oxygen, superoxide and hydrogen peroxide and uh, forms various forms of reactive oxygen that come around the bacterium to control the bacterium and to degrade the bacterium. So we can take a reactive oxygen stain, something that shows reactive oxygen, and you can see the bacteria inside the cells. Okay, and so this actually shows the bacteria in those root hairs using a reactive oxygen stain. And this is a yeast. Okay, and you can see the yeast, these little dots, these are the yeast inside the root cells, inside the root hairs. And you see them emerging. You can see older ones and you see younger ones here as they come out. They actually will come out of the tips of hairs, root hairs, as those hairs grow. So they're actually being processed through roots. They go in the root, growing root cells near the meristem. They go out the root hairs as the root hairs develop. And this is actually an alga. And you can see all these little things, those are bacteria around the alga. The alga can also, the algae, some algae can be taken into the root cells as well. And you see the bacteria coming out of the algae, all those little dots there, those are, and you see the alga losing the chlorophyll as well. So there is a little process happening that we didn't understand that was happening in roots, this little rhizophagy process, oxidative process. What we have found is that this is actually a cyclic process, that this is a root here, root tip here, root hairs here. You see at the root tip, exudates are produced, secreted, microbes come toward those exudates. They accumulate here. They're taken into the cells here, and then they will trigger root hair formation. These hairs won't form unless those bacteria are there. And as those root hairs grow, the bacteria are ejected from the tips. And you see these little dots? Those are bacteria coming out of those tips. And again, this is invisible unless you have a reactive oxygen stain or something that works. So this has only been discovered in the last 15 years. It's a very recent understanding that this process is happening. And let's see what else we have. This is just a little diagram showing that nice cartoon that a colleague of mine in Austria produced. But you see the bacteria coming in, triggering hair formation, and then going out of the hairs. Coming out of the hairs, for example, there. This happens at root tips. So plants that make a lot of root tips do this rhizophagy process a lot. And you can actually see this is a grass root, a corn root. All these little branches, those are the tips. So grasses and plants that make a lot of branch roots do a lot of rhizophagy and do this process a lot. So they're getting a lot of nutrients from. And in fact, if you grow, for example, a corn without microbes, it'll have these little tiny roots, little tiny root systems. You put the microbes there and the roots become big and branching. It favors root development, but it also favors shoot development. So it's not only the roots, but the shoots also are impacted and increased. This is an image that shows a root, branch root, and these are the bacteria that the plant is cultivating at the root, that tip. And it is taking those microbes in right in this zone. Okay, this just shows some of the bacteria inside the cells. And you can see this side over here. You can see it's where the bacteria are taken in, and over here, a bit further over here is where the bacteria are actually oxidized and nutrients are extracted from the microbe. And in that extraction process, what happens is the plant will oxidize the cell walls off of the bacteria. So the cell walls will become deposited inside the root cells. And you can see these angular structures. Those angular structures are the cell walls. This is stained with crystal violet, which is a bacterial cell wall stain. And you can see these cell walls, the darker areas coming off of the bacteria. And what it does is it leaves protoplasts inside the bacterial protoplasts inside the root cells. Okay, and these protoplasts go to. Oh, I should mention one other thing. Nutrients are carried on those cell walls. When the bacteria are in the soil, nutrients adhere to the walls. And so when the plant takes those cell walls off, it actually is taking nutrients with it. There's protein also in those cell walls. So this is an image that shows a bacterium and the little metals are adhering to it. All these little white dots. Okay, that just illustrates the point that these nutrients are adhering to the cell walls. So the walls themselves carry nutrients. But the cell walls come off of the bacteria and they form protoplasts. And you can see these dots now, these little orange dots, those actually are the protoplasts of bacteria. They may be rods on the outside. When they go in, they lose their cell walls. They become these protoplasts inside. And here you can see also, you can see they're dividing. You can see a chain of 3 over here, sometimes chains of 2 over 2 where they're dividing. They are protoplasts, but they're still alive inside those root cells. Okay, and of course this whole process is affecting the biology of the plant, causing plants to be resistant to stresses and acquiring nutrients and other effects inside the plant. So plant health has a lot to do with these microbes being absorbed into the cells and tissues of the plant. Oops. Okay, this is just another graduate student work, some more work, but you can see these are actually cells here. And these are root cap cells, and this is a bacterium that was stained, or rather tagged, with red something called M. cherry stain, which is a fluorescent stain. And you can see the bacteria, the little red dots. Those are the bacteria inside those cells as it scans through that tissue. Okay, the bacteria are taken into the cells through the walls. The walls at the meristems are thin and elastic, very easy for the bacteria to go through. We don't know exactly how they go in there. We think that somehow they're absorbed. The Australian group said that they think it's an endocytosis of some sort. But also we know that some of these microbes can produce cellulases and other enzymes to break their way to get in. So they get in, and then they get trapped in there because those cells continue to develop. And the bacteria then are trapped inside those cells. As the cells develop, the walls harden, and the plant will also take control of the bacteria and begin to manage the bacteria inside the root cells. Okay, the enzyme in the plant that produces the reactive oxygen is this one. This is NADP oxidase, or NOx enzyme, and it will take oxygen from the air. In roots, for example, the air in the root spaces and the soil spaces take that oxygen and turn that oxygen into superoxide. And that superoxide is highly potent form of reactive oxygen, and that's what the plant will use to oxidize these bacteria and control and manage the bacteria. So that's the enzyme that the plant is using to essentially control these microbes. And what the plant does will take a bacterium with a cell like this bacillus, these little rods, and will oxidize the walls off, and you get these little forms. These are called these little protoplasts are actually called L forms, and they're all well known. I'm not saying very much that's really new. I'm just putting together and telling you a different story about all of these uh, microbes and how they're working in plants, what's happening to them. Okay, the fungi work the same way. The plant will take fungi in, yeasts, oxidize the cell walls off, and produce these protoplasts. And they actually just have a different name. They're called mycosomes. And here they are inside. Inside, you can see they also are alive. Those mycosomes are alive. So a plant is not just a plant. A plant is a plant, its own cells, its own biology, its own genetics, with the microbes inside its tissues, inside its cells. Okay, and these microbes are important for plant development. Okay, for example, uh, the microbes themselves are necessary to make root hairs, and I'll show you that. They're necessary to make root branching as well. They're also necessary for the root to have the gravitropic response. We know that roots normally grow down with gravity into the soil. If you remove the microbes, they don't. They sometimes grow into the air. Usually they just grow on the surface like this. They don't grow down. So that whole response of the plant, the way it grows, the structures it forms, is dependent on these microbes going into the plant tissues. Okay, so this is actually a grass seed. This is we call it Bermuda grass. It's Synodon. It's common. You have it here. It's a common grass in this area. We remove the microbes, and you see you have the root tip here, part of the root here. You have little bumps here. Those are actually root hair initials, but without microbes there, they don't elongate. They don't grow. Same experiment where we put microbes back on, and you see now you have the root hairs forming on there. Immediately you have root hairs forming. Okay, and here's a bit further on, older part of the root, and you can see here, main branch, you see the long root hairs there. You can see little brown dots in there. Those little brown dots, those are the bacteria inside those root hairs. Okay, they are full of bacteria. They're not just root hairs. They're root hairs with their bacteria. These root hairs are actually microbe processing structures. So they're not simply absorptive structures, and I'll show you that. Okay, this shows a root hair from that experiment. You see the little dots in there? These are alive. These are alive. All those bacteria are alive inside that root. So living from the soil. This is tomato. Tomato. Here's where we use streptomycin, antibiotic, to remove the bacteria, and you see there's no hairs forming. Here we did not, and you can see the root hairs forming. Okay, this just shows the same thing. I don't need to reiterate again and again and again, but you see the bacteria accumulating in the tip. The bacteria in the tip trigger elongation of the root hair, and it grows. Without the bacteria, it doesn't happen. Okay, and this shows, I'll show here, you see the microbes here at the root tip, and you can see the microbes, bacteria coming out of this root hair tip right there. These are root hairs here. You can see the fluorescence there. Those are the bacteria coming out of those hairs. These hairs are microbe processing structures. They process these microbes. Okay, why does it cause growth? Well, we think it causes growth. We think it causes growth because they're producing hormones, because they're making hormones. And let's see if I can go to that. Some of the hormones that they make are these: ethylene, nitric oxide, and also nitrogen. Nitrate acts as a hormone. So those are developing. Those cause development. There's some other processes that also cause development that we're just learning about that trigger growth in the plant. So the microbes are triggering growth. How many minutes do I have left? Am I close to a couple of minutes? 5? How many minutes do I have? 5 minutes. Beautiful. Okay, beautiful. Okay, I will do that. Okay, because I want to show you a couple of other things before this is over. This is a sedge. It's growing in rocks here, and it doesn't have a lot of soil, but what it does have is a lot of microbes. And I will show you this. Here you go. Here you can see this is an image of a root hair for the sedge. You can see these brown things. This is with the reactive oxygen, and those are the bacteria there. And look here at the hair. This is living. This is a movie from the living roots. And you see how these hairs are working. They're processing these microbes around and around and around. You can see these shadows coming around. Those are the bacteria that are processing like that, going around. This is what these root hairs are actually doing with these microbes. And this shows the bacteria now in a hair. You can see what's happening. They're replicating. These protoplasts are breaking up, making clusters. So the plant is replicating the bacteria, and then it's ejecting the bacteria out of the tip. Okay, here you see a root hair. You can see the bacteria accumulating in the tip. And that bacteria in the tip produce hormones and causes the hair to grow. And when it grows, some of those bacteria are ejected out. And you can see here, you can see here, see how fat they are. These are full of microbes. You can see the microbes coming out. So when they come out, they reform their cell walls. And they go, if they have flagella, they go back into the soil, swimming and so forth. So, and this just shows a diagram that shows that the ejection process, every time you get a growth spurt, some of those microbes are ejected, some stay in, they're circulated around, they replicate, and some accumulate again, and then you have another growth spurt like that. So this kind of shows that you see a hair, you see where the bacteria accumulated, and each time you get a growth spurt. If we take the bacteria away, you get none of that. None of that. The growth is dependent on the microbes being present. And this is a tomato root hair. And you can see the tomato, you can see the ejections here. You can see the microbes are coming out of those hair tips, those hairs. Okay, the plants are actually using those microbes for their own development. And I will think I will summarize now. The process, this process gives plants not only nutrients, but it also makes plants develop. So they become larger root systems. They get more nutrients from those root systems. These microbes that they use go out into the soil, they'll affect pathogenic fungi. They'll affect fungi like fusarium that causes disease. They'll cause those fungi to be non-pathogenic, to be less virulent. Because they're in the plant would become oxidatively stress tolerant. And some of the disease, they can then tolerate diseases better. They can tolerate drought, for example, better. They can tolerate, chemical stresses, heavy metals better. So, in balance, in summary, plants actually are not plants. At least they're not healthy plants. They're not well nourished plants. Even if we're feeding them liquid fertilizer, they're better nourished and healthier with the microbes there. And so, as a bioresource, these soil microbes and these plant microbes isolated directly from plants can be useful for agriculture. And I think I'll leave it at that. I may be a few couple of minutes over. I apologize for doing that, Sereni. I'm sorry. Uh, how do you want to, do you want me to do questions or you want to wait till later for questions? No questions. No questions. Okay. Thank you so much. Thank you so much, everybody.
And our second keynote speaker is Professor P.B. Turney Pradeep Kumar. Professor P.B. Turney Pradeep Kumar is a proud alumnus of Rahula College, Mathura, and holds a BSc degree in zoology from University of Ruhuna. And he obtained his PhD in coral ecology from University of Kalmar, Sweden. He was the former head of the Department of Oceanography and Marine Geology and the former general manager of Marine Environment Protection Authority, Ministry of Environment, and also the former director for the Center for International Affairs, University of Ruhuna. He served in above portfolios for 7 years each, with active engagement in marine and coastal ecological management, administration, knowledge dissemination, research, local and international assignments, and consultancy for more than 25 years. At present, he is serving as a professor in oceanography attached to the Department of Oceanography and Marine Geology, University of Ruhuna, and his current portfolio is the Director General, Coastal Conservation and Coastal Resource Development, Ministry of Environment. He has published 35 peer reviewed research publications with the contribution to 5 books and 13 book chapters and more than 100 research abstracts. Professor Turney was a UNEP advisory group member and UNEP expert group member for the marine litter and microplastics, and also a SACEP expert member for marine litter management in South Asian seas for the preparation of marine litter management plan for South Asian seas region. At present, he is instrumental in policy development and on-site action and awareness campaigns on coastal and marine pollution control, plastic pollution including microplastics, marine protected areas, marine spatial planning, marine investment planning, spatial area management, and blue economic growth, management of marine industry in Sri Lanka. With this note, I cordially invite Professor Tani Pradeep Kumar to address the gathering.
Also added to the history of this faculty. And second one is, I think James, he did his speech through a vertical arena because he selected, of course, the plant roots and microbes actions there, and he went in depth to that. But as an administrator and as a researcher, I take the same, but I go horizontally. A big picture because it's the first symposium of this faculty, and as the vice chancellor and dean mentioned, this faculty is mainly meant for the national development. Therefore, how we can place this faculty in the national development and how our students focus their future and focus their research and focus their activities, and how this faculty and university actively engage in the national development is my main theme of my speech. If I can see my slides here. Yeah. So when I was asked to do the keynote speech at the very onset, I was a bit confused about what's the scope of this faculty. Therefore, I had to contact Surani and get the prospectus downloaded and read what you are teaching and what you are doing, right? Then I thought, okay, this is the area. So as per the understanding I took, I carved the topic, unveiling sustainable ocean wealth for future generations, bioresources for in the blue economy. So when we discuss about economies, we use colors when we discuss about economies. For example, we use brown economy, green economy, blues, golden, purple. Those the colors we use to describe various economies. For example, the brown economy. In brown economy, we talk about the fossil fuel like coal and oil gas, mainly focusing on destruction. Right? Then red economy, especially those linear. We use resources and we make trashes and the waste production. It's kind of linear economy. Then we talk about green economy. In green economy, we talk about focus on sectors like energy, transport, sometimes agriculture. Then the blue. In blue economy, we focus about the ocean, the waters, and even fisheries. What is happening with water resources? In actual term, what is blue economy? So we talk a lot about blue economy, right? So blue economy in very simple term, the use of ocean in more sustainable manner. Very simple, right? Right? Right. So we use ocean for food, very simply, fisheries. And we use ocean for energy. We use ocean for transportation and again extract minerals. And we use ocean for extract water, fresh water from saline waters. And we use ocean for leisure and we use ocean for health. But everything comes under the theme of sustainability. So we are in the sustainable development era, SDG era. So what is sustainability? In real term, the sustainability is a theme where we make a balance between social sustainability, sus- high good social life and sound environments and very sound economic development. So if you want to make a balance between these three, we can ensure the sustainability. So when we use ocean in more sustainable manner, what we have to do? We use the ocean, but always we ensure the health of the ocean. We ensure the ocean uses even for future generation. That's the ocean sustainability. In blue economy, we use ocean in more sustainable manner, right? So there are a number of uh, definitions if you browse the net, right, about what is blue economy is. If I take the first one, the World Bank one, the sustainable use of ocean resources for economic growth, as I mentioned you, and improved livelihoods and jobs at the same time while preserving the health of the ocean. So all blue economy is mainly focusing on these three major pillars. If you take up the ocean, so ocean wealth is 24 trillion US dollars. That does not encompasses the services and functions of our ecosystems, but other the physical wealth. Look at these four, the largest funds in the world. First one is the Norwegian Government Fund. This is Abu Dhabi Investment Authority Fund, Saudi Arabia Monetary Authority Fund, and China Investment Corporation Fund. Those are in billions, whereas the ocean economy that goes into trillions, right? So as an island nation, our future should be focused especially on this area where the high huge wealth is based. Therefore, ocean matters for us as an island nation. Ocean matters. When you, as a faculty of bioresources faculty, of course, you have to focus on land bioresources. But again, you have to focus much more emphasis on the ocean bioresources. Right? Why I'm saying this? If I look at this picture, I'll ask a simple question. Right? I always ask this: how many oceans we are having, sir? The dean, sir. How many oceans we are having? Five oceans. Yes. So we talk about five oceans, right? But in real terms, we have only one ocean, right? So everything is connected, right? In the ocean, everything is connected. If you go back to this picture, you can see it's Antarctica, and you can see all the other continents, but it's only one ocean, right? We have only one ocean, but number of continents, number of islands, and others. If you look at this picture from Pacific perspective, this Pacific Ocean, the largest ocean. So you will see barely very few islands. Or you will see Australia at Australia and New Zealand somewhere here. So everything is covered with the ocean. So ocean is the largest ecosystem in our planet, right? Therefore, when you focus about ocean, the bioresources come to ocean. Let's do diving and find out what are there and how we can use them for future development. The ocean is the largest ecosystem, you know that. So out of the whole, the hydrosphere, about 97% of water is there in the ocean. The 3% is the rest of 3%. Out of that 3%, one is in the soil as a soil moisture where those roots and bacteria works. The rest 1% in the air as air moisture. Still we are having still 1% of water for our own uses. And ocean, we have lots of ocean uses. I'm not saying those, right? But we use ocean for a number of our sustains. But we depending on our ocean. Why? We from our childhood we learn that we use oxygen for our own survival. But now with the current research, scientists have published that about 50 to 90% of oxygen coming from the ocean, from the surface photosynthesis, from plants, from ocean algae, from ocean planktons. Right? Therefore, ocean makes our world a livable place. Right? It makes our planet a livable place. So then I come to Sri Lanka. In our case, this is our ocean. Sri Lanka's, you know, jurisdiction extends from the land base into the o- the open sea that goes from here to about 200 nautical miles. So this whole ocean is ours. So when we plan Sri Lanka's future plans, so when we think about Sri Lanka's future resources, of course we have to think about that part also. This is about 8 times larger. That's horizontal expansion. But if you go to vertical expansion, look at this. So from coastline, again 200 nautical miles into the offshore. But if you go to depth, it goes to about 3,000 to 4,000 meters from ocean surface, from planktons and other larger charismatic animals, and animals living in deep waters. Again, our bioresources in our case, right? Therefore, we have to think about that. Not only bioresources in our jungles and other places, right? This area we have to focus on. So now I move to what are the ocean bioresources are. Those are fisheries, aquaculture, of course, you know, seaweeds, marine biotechnological resources, and coastal ecosystems. And fisheries, of course. So how we can develop fisheries resources for the future users. Look at this. These are pictures taken by me. Some are my pictures and some are taken by others. These are the fisheries resources you haven't seen in actual term, right? These are again paravagilian jacks. If you go down south, you can see them. If you dive, you can see those jacks. These are called spawning aggregations, right? That's where we use beach scenes and take them to the beach, right? These are our fisheries resources. So in the current world, out of whole fisheries and aquaculture production, that goes about 223 million tons. The capture fisheries goes to 92.3 million, and marine capture fisheries count for 81 million, right? So you can see here, capture fisheries is much more. The capture fisheries out of this 81%, 81 million is from ocean, right? The 11%, 11.3 are from the land, the maybe from fresh water. So it contributes a lot for our fishery production. You can see these are big trawling holes, not from the trawlers we are seeing in Sri Lanka, but to some mega trawlers, right? And the current status of this fishery resources, if I look it from here, you can see overfished stocks are about 37.7. So we have to do something from from research how to replenish these stocks is our duty, right? We need we have to do some research to increase that number from 37 to further up. And still we have 62 point of biologically sustainable resources where we are doing our fisheries activities at the moment. And what are the untouched or unexploited marine fishery resources in our ocean? You can see still there are deep sea fish resources are there still untouched. So if you want to do some research, you can find where they are and how we can catch them. And the mesopelagic fish. Mesopelagic is the area where the ocean depth goes to 200 to 1,000 meters. That area or the water column, right? The fish species, and again marine invertebrates, sea cucumbers, and again up to marine worms, and again seaweed. I come to that later, right? If you look at this graph, you can see it's 1972, I think. 70, it's 1974, and this 2021. You can see this is sustainable catches. This is unsustainable. It's about 10%. But by now, what has happened? Most of our fish catch or fish, industries unsustainable. They are leading into unsustainability, right? So then we have to understand how we can reduce this at the moment and how we can increase this sustainability practices, right? So how we can increase fisheries resources? For example, we can go modernize fisheries. We can think of go for digital management and data collection because now we are in the digital world. Right? We can think about vessel digitization, regulatory tracking, vessel tracking, and again curbing illegal fishing, and again infrastructure wise, harbor development, export standards. We put standards and we meet them. How to do that? And again sustainable aquaculture, culture-based fisheries, and shrimp and farming new techniques to find aquaculture. We started, we started we were young, so we disappeared. Taking class again. We were taking class, we were taking class in 1980s. Taking class began as a graduate from using that in Australia. It's a both period, sharing at all. Right? We are in which the government, the global demand increased, and export market opened, and government promoted shrimp farming, even in Sri Lanka. And there's also new and species like Lithopinus swanami and Penaeus monodon, though they were introduced. And what happened in Sri Lanka? We destroyed our mangroves. We converted our mangroves into huge, aquaculture land stands. So in that case also, it's a rush, yes, but still we did lots of economical damage, ecological damage. Now we are paying the price, right? And global production of trends at present, if you look at this graph, you can see this is total production, fisheries production from, and this is total aquaculture, and this is total wild fish culture, capture fishes from the ocean. In 2020, for the first time in the history, the aquaculture production passed this threshold, and now we have more cultured fish than wild catch fish, right? Therefore, we had to, of course, develop this culture-based industry. What happens to the those fish are maybe increase those aquaculture? Look at this report. Look at the report there. World this becomes 61 and say that the global tax farm species reduces focus time at the same time when you increase farm species, small scale fishes need protection. That's really intelligent and clear. Because we have very small scale expand farming. How we ensure our values. And children, of course, again, the first time I provide the third one, the lead is in the fisheries sector. Efficient resource use and reduce environmental impacts, and integration of new food security to the system, then we can convert aquaculture into more blue and more sustainable system. And again, we can introduce eco-friendly practices like recirculating aquatic aquaculture systems, RAS systems, and integrated multi-trophic aquacultures. I think aquaculture is a topic taught here in this course, IMTA, and reduce reliance on fish meal and fish oil, right? Again, go from linear to circular system in the aquaculture system. From aquaculture systems, technologies, outputs, then outcomes, then outputs and outcome become inputs again. The same system we use for our productions. Then with current world, that key new technologies where AI and smart technologies incorporate into aquaculture. For example, starting from GIS is well known, and spatial modeling is well known there, right? But the next step is IoTs, Internet of Things. So we save our data in the internet and use them in cloud forms, right? And again, AIs. For example, use fish AIs for fish growth and biomass estimation, and to ensure the fish feed optimization and disease detection. Use AIs. Those are the new technologies used in fisheries sector. And of course, you have to understand the difference between what is AI and what is automated system. These are two different things, right? AI, we use our data to get some predictions in AI. Look at this picture. So by taking the picture using AI, it determines the weight of this fish, right? We are not supposed to go and weigh the fish. The system automatically gives everything, and even the growth rates, everything we can get from AI. Right. Then cloud computing, right? Again, in that cloud computing in aquaculture and fisheries, real-time monitoring and alerts, the smart feeding systems, predictive analytics and AIs, and supply chain traceability. So look at this latest the publication from in 2025. Decision making support framework for aquaculture using multi-source data hub. I think in this faculty also you have to use AIs and databases for your future predictions and blockchain. Right? I hope you have an idea about blockchains. Anytime. Right, okay. Right. And the UVs, of course. I have to go now. Take a big rush now. These are about aquaculture. And then smart aquaculture systems and biofloc systems. I hope you are learning about. Then we have to understand how we use them. In Sri Lanka, we are still stepping into this area, especially in Trincomalee, using barramundi, the fish. But the big potential in Sri Lanka also, especially Japan and China, even Thailand, Taiwan. They are into offshore fisheries. Look at the offshore fish farms, right? In this case, you don't need to have a fine with. This sort of analyze system and we convert it into a natural environment. So we get our powers, the same time we encourage other animals to grow in space. And this is one of the systems. That's not aquaculture. Right? I will show this was. See resource. The next one, the CV. How we use CV blue economy. CV is main component users for green clothing doing one. So we can use series to now the product. And because we don't need any pollution, we can have now. We are not using because we use high qualification CDD culture for sustainable livelihoods, and no fresh water or fertilizers or land needed, help in carbon sequestration when we get the harvest, and contribute multiple industries. And again, therefore, in seaweed is again a key component in blue economy. Like this is something stats about seaweed production in at present in the world. And not only that, we can extract energy also from seaweed, right? So we can extract biogas, bioethanol, biodiesel, biohydrogen. I think this is an area we have to think about, right, when you see seaweed culture in Sri Lankan context. And benefits. Ah, I'm rushing. But again, we can use seaweed in tourism also, right? So that's another field we have to exploit when you think about Sri Lankan context, because we can have seaweed farming tourism, and seaweed spa and wellness tourism, and seaweed gastronomy and ecotourism in marine education using seaweeds. And the fourth one is marine biotechnology, right? Very quickly. So all the colors you see here, all the small chemicals they secrete is a resource for us. For, from your toothpaste to from your jelly, what you are eating, everything starts from there, right? This is a marine worm. It's polychaete worm, but this worm is very important extracting potential chemicals, right? This is a marine sea animal. All the colors are very much new colors are coming from the ocean, right? These color generations, right? So very important in bioprospectus issues, and genomics, right? Provide uh, resist for motion animals, right? And selective breeding and genetic improvement. If you look at this one, this is two coral, maybe I think coral crickets, and this is highly diverse. Therefore, after the disturbances, you can see still some are still living. It's monospecific. After disturbances, all dead. How to ensure how this diversity just like the genetic diversity. And again, hybrid trees can go far and give better stability and bacteria. You can take it now, but all these chemicals, all these drugs coming from the ocean resources, ocean animals, right? The industry is still growing. Therefore, we have still lots of opportunities to find new drugs, new chemicals. For example, in Sri Lanka, it is in Sri Lankan context, very few researchers are doing research on marine chemicals. The last one is coastal ecosystems and coastal ecosystem resources. Here, I don't talk about the importance of coastal ecosystems and coastal resources, but I will tell what we have done very quickly. Look at this coral culture, right? So these are done by us from our university, right? And this again, how this another culture we started in Marissa. This is how we started. This about 1 meter diameter large coral reef balls. This is now the situation, right? This thicket is about 3 meters in size, right? This is the techniques we use to ensure, ocean ecosystems and ensure fight against climate events, and again ensure more marine diversity in our coastal waters. This how we are. That's what we are doing at the moment. The next is to go for micropropagation. A lab is being built in Trincomalee at the moment, so the lab will be inaugurated in few months' time to go for micro and then use them in ocean environments to protect our coastline or increase marine diversity in all our coastal areas. This is what we are doing at the moment, right? These are micro propagation techniques we use in coral reef, right? This is what we are now focusing at the moment. Then, then the mangrove restoration. But in normal restoration, we plant mangroves and we expect about high carbon sequestration. But what we are doing at the moment with my agency is that we are focusing on using them into control marine coastal erosion. Some more special techniques, very simple but very effective. You can see these techniques. We use mangroves with other benefits to curb coastal erosion. Right? Some examples. These are actual notes. These are not from Sri Lanka, of course, but these techniques we are using now and go for next levels like this. Right? So these are new things. How to think positive. How to think new. How to think more futuristic way. In very short term, what are the economic benefits of blue resources? Bioresources. Those are bioresources contribute to the blue economy, of course. Food production, employment, export earnings, pharmaceutical development, biotechnology, innovation, renewable energy, ecotourism development, and carbon sequestration, of course, is well known. But these are emerging opportunities are there. Sport and precision aquaculture, seaweed farming for carbon neutral industries, marine bio refineries, blue biotechnology, coral restoration, nature-based climate solutions, circular bioeconomy, and sustainable aquafield development. Right. So what we have just learned is that we have a resource. We have a huge marine bioresources are there, right? Then how we can use them in a more productive way, and still we have a big opportunity, right? So when we use that opportunity, we have to always ensure the sustainability. If we can do that, and if we can focus for that, if we can think about future generations, I think this faculty will be the great, cradle to take them over and ultimately support our Sri Lankans and ultimately support our poor people who are still waiting for to some benefit from our ocean environments. With that, I conclude my speech. Thank you.
Not expressing it is like wrapping a present and not giving it. We witnessed two insightful and inspiring keynote sessions. Professor James White joined us all the way from the States to enlighten us on a very important topic, the role of soil microbes in crop production, addressing plants and microbes, dominant components of the terrestrial environment. Professor Turney Pradeep Kumar enlightened us on another timely, important topic, bioresources in the blue economy, addressing the aquatic environment. Together, they have addressed the importance of bioresources of both aquatic and terrestrial ecosystems for a sustainable blue-green economy. To express our gratitude, we prepared a small token of appreciation. I cordially invite Senior Professor Pradeep Jayavira, Dean of the Faculty of Urban and Aquatic Bioresources, to hand over the tokens of appreciation. I cordially invite Professor James White from the State University of New Jersey, USA, to accept his token of appreciation. Next, I cordially invite Professor Turney Pradeep Kumar, Director General, Department of Coastal Conservation and Coastal Resource Management, to accept his token. Thank you, sir. Over to you, madam. Thank you very much for the previous keynote speech, Professor Turney Pradeep Kumar. And now we came to the online keynote session. So we have our keynote speaker, Professor Alabakash, Associate Professor at the Center of Excellence in Molecular Biology, University of the Punjab, Pakistan, as the keynote speaker for ARCOP 2026 joining online. Dr. Bakash is an internationally recognized plant molecular biologist with expertise in plant genetic engineering, genome editing, RNA interferences, and development of climate resilient crop varieties. His research has significantly contributed to modern agricultural biotechnology, particularly in improving crop productivity, stress tolerance, and resistance to pests and diseases. Over his academic career, he has published more than 200 scientific papers with over 2,000 citations and has an H-index of around 30, reflecting his strong research impact in the field of plant biotechnology. Dr. Barkash currently leads the Biotechnology Laboratory at CEMB, where his research explores genome editing, RNA based technologies, and molecular breeding approaches for developing climate smart and resilient crop varieties. Although joining us virtually from Pakistan, we are honored to have him share with his valuable insight and expertise with our COP 2026. Over to you, Professor Alabakash. Yeah, thank you, sir.
So, good morning. Can you hear me?
Yes, Professor Alabakash, I already gave an introduction to but the computer was muted. I think you didn't hear. So
Okay.
you can, I over to you to start your presentation, Professor Alabakash. Really sorry, extremely sorry about that. The computer was muted when I introduced you to the gathering.
That's fine. No problem. So, so I can start now. Thank you. Okay. Good morning, everyone. Distinguished professors, researchers, students, and guests. It is a great privilege for me to be invited to this prestigious conference organized by Faculty of Urban and Aquatic Bioresources, University of Jawadharpura, Colombo. Thank you so much. I'll talk about some of research work which we are doing at Center of Excellence in Molecular Biology, and there I'll discuss how genome editing can contribute to the development of climate smart crops. I'll take tomatoes as a case studies. And, the climate change is creating unprecedented challenges for us, for all the scientists, the rising temperatures, emerging pathogens, irregular rainfall patterns, increasing food demands. These all requires innovative solutions. And in recent times, genome editing has emerged as a powerful technique to develop smart crops. Thank you so much, and I'll go through the slides. But before that slides, I would like to briefly introduce my institute, University of Punjab. I belong to University of Punjab. This was established back in 1882 when British were ruling this part of subcontinent. And over the past century, the university has produced thousands of scientists, educators, policymakers who have contributed significantly to national development. And the university has a strong commitment for research and development, and particularly in agriculture, biotechnology, and life sciences. This is situated in Lahore city, and I belong to Center of Excellence in Molecular Biology. And this center was established in 85, 86. And you can imagine at that time, molecular biology, biotechnology were in infancy. They were developing in different parts of the world. At that time, the center was established and to strengthen Pakistan's capabilities in molecular biology and biotechnology. And over the years, CEMB, the Center of Excellence in Molecular Biology, has become one of the leading biotech institutes in the country and Asia. So the institute has different research programs, work in agriculture biotechnology, in environmental biotechnology, health biotechnology. So it's a there is a cluster of different labs, and among the plant laboratories, I take care of seed biotech lab. And when I talk about the seed biotech lab, we work on different important research themes. Basically, it's a cluster of different labs, and we deal with the insect resistance, weedicide resistance. We develop dsRNA-based pesticides, and the value addition in crops is another target. Because we develop different GMOs, and so there is always a necessity to go through the risk assessment studies. So we have a full-fledged animal house where we rare different animal models and keep on our biotoxicity studies on these animal models. So besides that, we also work with the virus resistance and secondary metabolite engineering. So these are the areas, the themes where we use all our efforts. And for recently, like I told you, to accomplish our targets, we use different molecular tools like RNAi and genome editing to address these challenges in crops. And we are a generation where we have come across severe problems and changing from the climate, like I mentioned in the start, and the insect pest and diseases. That's another problem associated with the climate changes, and the water, another another area which is of extreme importance in today's time. And another challenge we have is the growing population. Today, it's another challenge, and the less arable land. The way the urbanization is increasing in many developing and countries. I think it's swallowing our agricultural land, and that's another major concern. And besides all this and associated of all these, we have to breed, we have to come up with the crops, with the plants that are climate smart, tolerant, and resistance to drought, heat, salinity, and submergence. And these are the challenges which we, as a young researchers, have to follow, and we have to come across, like I said, with the innovative solutions to fix those. Uh, like I said, the genome editing basically, like we have witnessed the success of talents and ZNFs, and since 2013, we have CRISPR-Cas with us, and CRISPR-Cas originated as a natural immune system in bacteria, and we know that bacteria are constantly attacked by viruses. To defend themselves, they evolved CRISPR system that can recognize and cut invading viral DNA. The scientists realized that this natural mechanism could be adopted as a programmable genome editing tool. Genome editing system consists of primarily of Cas9 proteins, which acts as a molecular scissor, and then the guide RNAs basically which directs Cas9 to make a specific cuts on a specific DNA sequences. So together they create highly precise cuts at targeted locations within the genome. Once the DNA is cut, the natural repair mechanisms come into play. They generate mutations and allow targeted modifications. So that's how the system makes it very easy and very popular. And why it's a popular? Several reasons. The firstly, it's relatively inexpensive in today's time compared to the previous systems like ZNFs and talons, zinc finger nucleases and talons. And secondly, it is highly efficient and it's versatile because any genes can be targeted. And now we have a lot of different tools where we can avoid the off-targeted mutations. And it significantly reduces the unwanted random mutation compared with the conventional mutagenesis. And most importantly, genome editing can produce changes that are indistinguishable from naturally occurring mutations also. As a result, this system has rapidly become the preferred genome editing platform worldwide. And if you look at the figure, this summarizes how the genome editing works, and this workflow journey involves, designing of guide RNAs, the development of the CRISPR constructs, delivery into plant cells through different methods, through agrobacteriums, particle bombardment, RNPs, and when the complex gets entered into the plants, you go and screen your edited ones. And the beauty of this system is even you can remove in today's time you can remove the plasmid's backbone, and only the mutations will get inherited to the subsequent progenies. This is this is the area where we spend most of our time in our in silico studies and finding our targets. And once our targets are defined, then the rest of the procedure becomes a sort of routine process in a laboratory. So I'll give you, like I said, I'll give you a case example of tomatoes. Tomatoes are I must say, it's one of the most important vegetable crops worldwide, and and also it serves as an excellent model for functional genomic studies. So, and like I said, it's the most important vegetables, and the China is leading with 67% million tons, and 37, it contributes to 36% of the fresh and processed tomatoes, followed by India, Turkey, US, Italy. So one of the widely consumed vegetables, and also it's a good model plant in labs. So it's exposed to various bacterial diseases and comprising of bacterial wilt, canker, soft rot, spec, and these diseases are constant threat to the crops and leading to overall productivity. 20 to 40% losses have been reported. Cost losses are also there. So and I believe that genome editing is especially the CRISPR-Cas are one of the best tools against the viruses and bacterial and fungal pathogens. And for this, we to we used the system to combat the bacterial pathogens using the CPL3 genes. And to identify the potential targets for genome editing, we first explored transcriptomic and genomic information related to plant pathogen interactions. And one candidate that emerged was CPL3 gene. The CPL3 encodes a CTD phosphatase like protein, a nuclear phosphatase, in fact, that regulates gene transcription by dephosphorylizing the C terminal domain of RNA polymerase II. Rather than directly recognizing pathogens or environmentally signals, CPL3 acts as a transcriptional regulator, controlling the expression of many downstream genes involved in the development and stress responses. The genes consist of, it's 11 exons, 11 coding regions. So we identified the coding region 1 and 5, exon 1 and 5 as the best targets. And we also did some protein analysis, and the protein modeling revealed several functional domains and protein interaction pockets. So protein pockets are important. Protein pockets are important because they often indicate regions involved in signaling, and that's why, and the other regulatory interactions. The transcriptomic evidence suggested that CPL3 expression changes during pathogen attacks. The literature also suggested CPL3 may play a significant role during stress response. That's why CPL3 emerged as one of the candidates and one of the negative regulator of the immunity of the plant. That's why we decided to knock out these genes and to see its effect in plant pathogen interaction. So the first experimental step, like I showed you earlier in workflow, was the identification of the target genes. And we amplified two exonic regions, exon 1 and exon 5, uh, keeping in mind the domains and the functional targets. So the fragments were amplified, and they were cloned in a bacterial vector and further sent for the sequencing to characterize. And like you can see, and the confirmation was done like the molecular tools like PCR restriction analysis. And PCR restriction analysis, they showed that both of the targeted internal fragments, they have been cloned in vectors, and they were sent for the Sanger sequencing, and the sequencing further validated the sequences which we found from the study, the already available literature. So this step was essential because accurate information is required for designing highly specific guide RNAs. And besides that, to understand the biological role of CPL3, we evaluated expression under plant pathogen interaction in pathogen stress. 3 cultivars were used: Rio Grande, Sazlija, and Falcon, and the measurements were taken at 24, 48, and 72 hours post-inoculation. We monitored antioxidant enzymes like ascorbate peroxidase, catalase, and salt. We also measured the oxidative stress indicators, H2O2 and MDA. So the biochemical and gene expression, and also we performed the qPCR analysis of the stressed plants. So the biochemical and gene expression analysis indicated that under biotic stress, CPL3 gene is highly expressed, and H2O2 and MDA level is also higher as the time progressed. So on the other side, ascorbate and catalase and salt activity was less in Rio Grande compared to the other genotypes. And keeping in view the susceptibility, this genotype was selected for further studies. And it also established that during the stress, CPL3's expression is enhanced. So, like I said, then once the sequence was confirmed and it was characterized, and then the guide RNAs were designed, and these guide RNAs were inserted into PKSC401. It's a commonly used CRISPR vector that contains Cas9. That is like monocot optimized, and once the guide RNAs were designed, what all the other factors were kept in mind while designing guide RNAs. These guide RNAs were synthesized and annealed together to make a duplex, and this duplex were further integrated or cloned in PKSC401. And like in our studies, uh, we used two vectors, like I said, PKSC401 and Osakabe vector. So, and we had two guide RNAs, guide RNA 1 and guide RNA 2. This was of exon 1, and this was one exon 5. And once the guide RNA were cloned into expression vectors, they were conformed by qPCR, by colony PCR, normal PCR, restriction analysis, and once these things were conformed, and earlier the tomato hairy root regeneration transformation experiments were conducted. Roots were conformed by PCR using the amplicon sequences, and further after transformation, we assessed the editing efficiency. The high resolution fragment analysis allows the detection of small insertions and deletions. The appearance of multiple peaks indicates the presence of edited alleles. These techniques provide rapid screening of large number of plants before screening confirmation. So our results showed that the Osakabe vector showed maximum editing efficiency in hairy root transformation. So besides hairy root transformation, we also performed the agrobacterium mediated transformation, Rio Grande stable transformation by using the optimized protocols, which we keep on doing in our labs. The PKSC4 was used for that purpose, and the cotyledon nodes that were used and explants, the regenerants were obtained, and that were further confirmed by different PCR analysis and amplicons analysis, which I'll show you. Like this was the restriction analysis of the exon 1. There was a unique site in the amplicon, the targeted amplicon, that really helped us to understand whether is there any mutations or not. So this restriction digestion analysis confirmed the successful editing of exon 1. Several plants showed altered digestion pattern compared with the wild type controls. These differences indicate mutations generated by Cas9. Some plants exhibited heterozygous mutations, suggesting successful modification of one allele. So likewise, again, the same was used for the exon 1. That basically helped us to understand that these individuals, plants, they are mutated one and they carry the targeted mutations. So these results confirm successful editing at the CPL3 site. So this just confirmed the mutations, and further were also these amplicons were also further sequenced, Sanger sequenced, and that further confirmed. And the important thing, ultimate goal is not just simply to create the mutant plant. We also need to improve the plant performance. So these edited plants were challenged with the Pseudomonas singer, and we measured leaf fresh weight and bacterial population growth. The difference between the wild type and the edited plants indicate the altered disease response. Basically, this is the wild type, and these are the edited ones. So this demonstrates that, and here you can see that the fresh weight and in wild type is reduced compared to the other ones. And likewise is the case with the bacterial count. So basically, this established the fact that CPL3 has a measurable role in host pathogen interaction. We further, to validate the gene functions, expressions, we perform different assays, like we saw the expression of CP exon 1 in edited plants in comparison to the control ones, of exon 5. And then we also looked as a marker gene, the PR1, to see the effect on the defense related proteins. So here you can see the phenotype. This is a wild type. Here, the wild type, you can see the most affected one, and these are the edited one. So basically, the changes in PR expression, the changes in PR expression indicate the editing CPL3 influences defense signaling pathways. So, and if you compare, if you look here, you can see the reduced transcript levels of exon 1 and 5 in mutated plants. So thus the molecular evidences support the phenotypic observation obtained during the pathogen stress. So these results were published in multiple biology reports. Just the characterization part, the rest of the part where we are doing all the phenotyping analysis, we are that research work is still under manuscript preparations and will be published in another journal. So, in short, if I conclude, I would say that we successfully identified and characterized CPL3 genes, and the transcriptomic and biochemical analysis suggested a role in stress response. CRISPR-Cas9 editing enabled precise modification of the targeted genes, and our functional studies demonstrate that CPL3 influences defense-related response in tomatoes. These findings illustrates how genome editing can accelerate the development of climate-smart crops with improved resilience. Yes, here I would also like to conclude, like, yes, genome editing can be helpful to develop climate smart crops. Of it's a good tool for the breeders, but there are many challenges like pending related to the regulatory issues and other compliance. And we need to see how faster they can really be helpful to us to combat the climate challenges ahead. So this research was. I would like to acknowledge our collaborators in Turkey and also in Italy, in Sphere, and Erasmus Plus. Our students they just uh, he completed part of the Hilly Road transformation in Korea. And I'm also grateful to the research team of Seed Bartic Lab, which are the main pillars of the research work. So also I acknowledge the support of our University of Punjab and Center of Excellence in Molecule for these studies. And now I'm ready to take any questions, and I'll be grateful to you if you can write for any queries and for any potential collaboration with us. Thank you so much.
Thank you very much, Professor Alabakash. We are not, since this is a keynote speech, we are not asking any questions from the audience. And I would like to thank you for your very insightful presentation and sharing your knowledge with us. And also, I'm really sorry for the technical difficulties that we faced today, and you have to wait a lot of time with us. And thank you very much for your presence here today. And we are great to have you here. Thank you very much.
Okay, welcome.
Thank you, Madam. That concludes the keynote sessions. We've reached the end of inaugural proceedings of the Annual Research Conference on Bioresources 2026. To conclude this session, I take this pleasure in inviting Dr. Udari Ratnatunga, Secretary of this conference, to propose the vote of thanks.
Okay. Good morning, ladies and gentlemen. As we come to the conclusion of this inaugural session of the annual research conference on Bioresources 2026, it is both an honor and privilege for me as the conference secretary to deliver the vote of thanks. Today marks a truly historic occasion for the Faculty of Urban and Aquatic Bioresources as the first ever annual research conference of our faculty. ARCOV 2026 represents a significant milestone in our journey towards the academic excellence, innovation, and scientific discovery. First and foremost, I would like to express our sincere gratitude to our chief guest, Senior Professor Patmalal Manike, Vice Chancellor of the University of Sri Jawadanapura, for gracing this occasion and delivering an inspiring address. Your visionary leadership and unwavering commitment to the advancing research and higher education continue to motivate us and guide us. We are deeply honored by the participation of our distinguished keynote speakers, Professor James White from Rutgers University, the University of New Jersey, United States of America, Professor Attorney Pradeep Kumara, Director General of the uh, Department of Coastal Conservation and Coastal Resource Management, and Professor Ala Baksh from Center for Excellence in Molecular Biology, University of Punjab, Pakistan. We sincerely thank you for sharing your valuable expertise, experience, and insights, which were enriched this conference and inspired our research community. I would like to extend my deepest appreciation to our dean, the conference chair, Senior Professor Pradeep Jayavira, whose vision, leadership, and continuous guidance were instrumental in transforming the idea of ARCOG 2026 into reality. His unwavering commitment to foster a vibrant research culture within the faculty has been the driving force behind this landmark event. My heartfelt appreciation also goes to Professor Surani Chaturika, conference coordinator, whose exceptional dedication, meticulous planning, and tireless efforts have been central to the successful organization of this conference. I would also like to acknowledge and thank all the deans, the registrar, head of the departments, academic staff members, researchers, and students who have joined us today. Your presence and support reflects the collaborative spirit that drives research innovation and academic excellence within our university community. A special note of appreciation is extended to our valued industry collaborators and partners whose engagement strengthens the connection between academic research and real world applications. We gratefully acknowledge Dr. Nishant Nanayakara, Chairman, Devara Group, Dr. Sanath Hettiarachi, Chairman, the National Aquatic Resource Research and Development Authority, and Mr. Anuradha Ranasinghe, Agricultural Innovations Manager of Dilma Ceylon Tea Company. I would also like to express my sincere gratitude to our administrative team. Special thanks to our former Senior Assistant Registrar Indu Malalgoda, and also wish to thank our current Assistant Registrar, Ms. Madhushani Purnima, for their commitment and professionalism in enduring the success of this conference. Our special appreciation also extended to all non-academic staff and also the support staff members whose invaluable contributions, hard work, and unwavering commitment played a vital role in the successful organization of this event. I would also like to acknowledge the remarkable efforts of the organizing committee, program committee, session coordinators, session chairs, and session evaluators who worked diligently over many months to coordinate every aspects of this conference. From scientific programming and technical session to evaluate, logistic, media coverage, hospitality, and participant engagement. Your collective efforts have made this inaugural session a resounding success. Special thanks also extended to the proceedings editorial committee and editorial board led by Professor Surani Chaturika as editor in chief, and Dr. Anusha Ekanayaka as managing editor, together with all associate editors. Your dedication and professionalism ensures the successful compilation and publication of the conference proceedings, creating a valuable scholarly record of the conference research presented at ARCOP 2026. We are also grateful to the National Science Foundation of Sri Lanka for supporting this conference through the support scheme for scientific meetings and events, and to the University of Sri Jawadhanapura for its continued financial support in promoting research and academic excellence. I would also like to recognize and congratulate the first graduating batch of the Faculty of Urban and Aquatic Bioresources. Your achievements symbolize the growth, progress, and success of this young faculty, and we're delighted to celebrate this historic milestone alongside you. Finally, I extend my sincere gratitude to all presenters, reviewers, participants, volunteers, media partners, including Japla Frames and Japla Media, and everyone who contributed in numerous ways to make ARCOP 2026 a success. As we celebrate the successful inauguration of ARCOP 2026, we are not merely concluding a ceremony. We are marking the beginning of a new chapter in this research journey of Faculty of Urban and Aquatic Bioresources, University of Sri Jawadhanapura. May this conference serve as a foundation for many more years of scientific excellence, collaboration, innovation, and impact. Thank you all for being part of this memorable occasion. Thank you.
You, madam. With the hope of practicing what we learned here today in real life, because actions speak louder than words. It is time to conclude inaugural proceedings. As Professor Turney highlighted that we have only one ocean, similarly, we have only one planet Earth, and there's no planet B. Therefore, let us leave greed aside and share resources equally and explore urban and aquatic bioresources towards a sustainable blue-green economy. Signing off as your host this morning, I'm Minil De Cruz.
And I'm Ritvik Ajay Saran.
Thank you and have a pleasant day.